Hello friends, welcome back to Shiva School, earlier known as the Knowledge Institute. Hence, for the Knowledge Institute YouTube channel will be referred as the Shiva School. Okay, so I am Pratap Bhosle. Welcomes you to our earlier zoology course. Okay, so we have all started with the cell biology chapter. Okay, slowly the speed of this course will also going to be increased. Okay, so but today our fourth lecture is there, lecture number 4, and today the chapter is lysosome. Okay, so I have already launched our website, that is shivaschool.co.in. Okay, but the contents are not fully updated, so I will inform you on the Telegram group and the YouTube channel about when the website is fully ready. Okay, then for those who are attending this lecture for the first time, okay, for those I would like to inform that I have already published my book on zoology UPSC question papers. It has 22 previous year question papers which I have sorted according to the micro point in the syllabus. Okay, so it will be useful for you. So let's start with the next lecture, okay, lysosome. So we have already seen about the cell membrane and all the fluid mosaic model and all that. So today is the lysosome. So the lysosome basically is an acidic organelle, which is a membrane-bound vesicle containing the enzymes which degrade the waste or foreign particles or foreign dead bodies, whatever the waste or harmful constituents get inside the cell. Okay, the lysosomes basically degrade, digest, and then expel them, all of them, outside the cell. Okay, so this is the basic structure of the lysosome. It is a single membrane structure. It has all the enzymes and proteins inside it, and on the membrane there is a V class proton ATPase pump, transport pump, and another one is the chloride ion channel. What these two basically do is that these V class proton pumps just maintain the pH inside the lysosome, acidity inside the lysosome. Okay, and the chloride ion channel maintains the chloride ion concentration inside the lysosome. Okay, so these are the two types of pumps or channels on the lysosome membrane. Okay, and there are almost 50 acid hydrolysis and other types of proteins inside the lysosome. Okay, so this is about the lysosome structure. Then we will go for the point the formation of the lysosome. Okay, so the formation is through several steps. Okay, and or several intermediate bodies. Okay, so there are many kind of intermediate bodies like early endosome, late endosome, multivesicular bodies, phagolysosome. So all are generally referred as the lysosome interchangeably. Okay, so there is no fixed boundary which could say that the lysosomes are only this one, and multivesicular bodies or the late endosomes are not lysosomes. They are function and they are referred interchangeably, or intermediate vesicles also. So we will going to see one by one. So let's say how the primary vesicles are formed. Okay, this is the rough endoplasmic reticulum and this is the Golgi bodies. Okay, so the basic origin of the lysosome starts from the rough endoplasmic reticulum, and what happens is that the rough endoplasmic reticulum synthesizes collected all the enzymes which are needed for the to be inside the lysosome. Okay, and the cisternae get broke off from the rough endoplasmic reticulum and reticulum endoculum, and it is just transferred to the Golgi bodies for the further processing. Okay, so these are what called as the primary vesicles. Okay, primary vesicles which have the enzymes which are required for the lysosomal activity. Okay, and these are some kind of points which I will likely to mention in the lecture only. Okay, so that the student will attend the lecture and not only read the notes and go visit it. Okay, so you need to attend the lecture because there will be some additional point I will be taking for the lecture. Okay, So these are the primary vesicles which are with the enzymes. Okay, then they are further packaged to the Golgi bodies. Now Golgi bodies is the final processing of this lysosomal or the primary vesicles, and when these vesicles get out from the Golgi complex, they are referred as a primary lysosome. Okay, so this is called as a formation. Okay, how they are origin, how they are originate or the form. Okay, then but this process occurs with the possibly postulated to be for two ways, right? One is with the involvement of the Golgi complex, and another is without the involvement of the Golgi complex. Okay. In some cells like neurons and exocrine pancreas, lysosomes are formed from enzyme-rich endoplasmic regions, but without Golgi complex involved. Okay, so just remember that point. And the lysosomes are formed when endocyte materials fuses with a late endosome or enzyme degradation begins. Okay, so next part is how the early endosomes are. Okay, so you can call it, there are three basic components or the basic phases of lysosomal life cycle, like the primary lysosome, early endosome, and the late endosome. Okay, these are the main important parts of the lysosome. So the early endosome is what? Early endosome is, these are the endosomes which are present near the cell membrane. And what is the basic function of them? They take part in the receptor mediated endocytosis. Okay, this plasma membrane has a kind of receptors which are responsible for the form that pouch, and the ligands get endocytosed in that pocket. Okay, but after that they come inside and they fuses with the early endosome. And what is the early endosome? Which is nothing but the formed out of those endosomes only. And when the multiple endosomes come together, they forms a structure like this, and this is basically present near the cell membrane only. Okay, they are not a deeper they are not present deeper inside. Okay, so these have the acidic pH and ATP, which are maintained by the ATP driven proton pumps. And what the early endosome do is that they just uncouple the receptors and the ligands from each other. Whatever the ligands which come from the outside and they have attached to these receptors, these early endosome uncouple them, and therefore they are also known as compartment for uncoupling of receptor. That is what? CURL. Okay, compartment for uncoupling of receptors and ligands. Okay, so the uncoupled receptors go back to plasma membrane and ligands to late endosome. Okay, so whatever the endocytosed ligands are there, they are then transferred towards the late endosome. Okay, after the early endosome comes into contact with the primary lysosome and all that. But the remaining receptors are there, they are transferred back. These receptors which are present here, they are transferred back to the plasma membrane. Why? Because they will function again and they will further endocytose the external or the foreign ligands into it. So that's why the receptors are transferred back to the plasma membrane, but the ligands which are endocytosed, they are transferred to the early endosome for the further digestion or the processing, right? So this is how the early endosomes work. Then the next is the late endosome. They are also called as the intermediate compartment. Okay, and it has a pH less than 5.5 acidic, and it what contains basically lysosomal hydrolysis and lysosomal membrane proteins. Okay, so this is we can call as a complete functioning unit of a lysosome, right? It has all the required lysosomal enzymes, proteins, and all the digestive, what are the foreign particles or the foreign bodies which are also going to be digested inside. So this is a complete functional body, late endosome, right? And both are formed in rough endoplasmic reticulum, transferred to the Golgi complex for processing, and then separate vesicles transferred to the late endosomes. Once all the enzymes are delivered to endosomes, these can finally be called as what? Lysosome. Okay. Then the next is now we have gone through these. What are these early endosome? What are the primary lysosome? And what are the late endosome? Now there are some or we can call it the functions of these lysosomes which are there. Okay, and what are these functions, or what will the fate of these lysosomes or the late endosomes? This is explained by the three processes mainly. Okay, and what are those? The first process is obviously the endocytosis, which we have already covered. And the next one is what? Phagocytosis, and the next function will be what? Autophagosis or the autophagy. Okay, so these are the processes by which the contents or the foreign bodies or the antigenic bodies come inside the lysosome. Okay, and by the process of exocytosis, all of the what is going to be formed after the processing or the digestion of these particles, it is going to be released to the exocytosis. Okay, so let's go for one by one. The first one will be the phagocytosis. What happens in the phagocytosis is that the plasma membrane form the pouch of where the foreign or the microbial bodies come inside to the contact of the plasma membrane, and it just engulfs it, it just phagocytoses it, right? Once it phagocytoses it, it forms a phagocytic vacuole inside the cytosol of the lysosome. Okay, and this phagocytic vacuole then combines with or get attached with the early endosome and the enzymes in the primary lysosomes, and finally it will form what? Phagolysosome. And why it is the phagolysosome? Because it is formed after the phagocytic vacuole is formed, although it is formed by the process after which is followed by phagocytosis, right? Then this phagolysosome, then the entire action on these foreign or the antigenic bodies and the microbes happens in this phagolysosome. It is just degraded, and finally it forms what? Residual bodies. It is just completely digested or completely processed final waste material, and it is what it finally called as. It is called as finally a residual body. Okay. Then the second process is endocytosis, which have already gone through that. The soluble macromolecules taken into the cell by invagination of coated beads, right? So these receptors at the plasma membrane helps, because it has to have a kind of receptors to get endocytose the foreign particles, okay, or get the foreign soluble macromolecules inside it. Okay, and once the endocytosis is occurs, it forms the early endosome, late endosome, and further it goes to another body called as vesicular bodies. Okay. And this multivesicular body is nothing but which is formed after all this, like late endosome, early endosome, lysosomes, all kind of vesicles are part of these multivesicular bodies. So that's why it is called as multivesicular bodies, because it is the process after all the process of these vesicles, like endosome, lysosome, and late endosome. Okay. But after the multivesicular bodies also finally goes to this residual body phase. Okay, because once all the enzymes deactivate all the material inside it, all the whatever the foreign particles or the macromolecules are inside it, then it becomes a kind of inactive body, and it finally again forms the residual body only. Okay, so this is the endocytosis. Then it is the autophagy. Okay. The dead organelles and bulk cytoplasm are delivered to lysosomes through the autophagic pathway. Right now we have gone through that. What will happen with the soluble macromolecules come? What will happen with some antigenic particles come, or some foreign particles which are needed to be digested comes to the lysosomes. These processes are important for all these kind of processes. Okay. But what will happen if the mitochondrion inside that cell has to be digested or has to be discarded? So at that time, the rough endoplasmic reticulum plays an important role here. Okay. And what role it plays? It forms an autophagic vacuole. Okay. So we have already seen that the primary vesicle is formed out of the rough endoplasmic reticulum, right? And again, this autophagic vacuole, the membrane of this autophagial vacuole is formed out of the extended part of the rough endoplasmic reticulum. Okay, so it just autophagized that mitochondria, and it forms the autophagic vacuole. Now, once the autophagic vacuole forms, it just attacked or combines with this late endosome, primary lysosomes, and it finally forms what? Autophagolysosome. Because it has the autophagolysosome has all the lysosomal enzymes, the enzymes present in the late endosome and all that. Okay, so once this process of digestion or this process of all the treatment of these waste particles or the breakdown of this dead mitochondria is complete, this autophagolysosome also get transformed to a residual body. Okay, so finally all these almost processes end in the residual body. Okay, and the residual body is the kind of body which has not active lysosomal enzymes in it. Okay, it is just an end product of all this process. And once it is formed, it is obviously not of any use to the body. So it is then exocytosed, or it is the content of this residual body is expelled from the cell by the process of exocytosis. Okay, so this diagram we can say this is the origin of the lysosome, and all of these are what? Functions of the lysosome. Okay, then all of these phagolysosome and phagocytic vacuole or the autophagic vacuole, they are also called as secondary lysosomes. Okay, so this I have just given them the different terminologies used to them, right? That like the autophagic vacuole is also called as cytolysosome, right? Or the residual body is also called as telolysosomes, post-lysosome, or dense body. Okay, you just have to remember all these synonyms. Okay, so the question might be asked with the synonyms only. Okay, so these are the structure of all these vesicles, and we should just differ by few points. Okay, so and how these are formed I have already explained. Okay, so let's move to the next part, that is the main functions. As I have said, this can also be the part of the lysosomal function. These are also the part of the lysosome function, but along with that there has to be an emphasis on the lysosomal enzymes when the question has been specifically asked about the enzymes of the lysosome. Okay, so all of this I have already explained in that phagocytosis and all the processes like the breakdown of foreign material, like degrade transport proteins, making their contents available to the cell, their digestion, their organelles and extramolecular molecules, right? Degrade extracellular material, sometimes play a role in program embryonic development by destroying water waste tissue. Okay, and the degrades polymers into their monomeric subunits. Okay, so these are all the enzymes. Okay, almost the few examples. You don't need to mug up all this. You just can give any one example of any enzyme types, like the acid lipase acts on lipids, acid DNase, acid RNase acts on nucleic acid, like this glucuronidase or N-acetyl galactosamidase acts on these complex lipids, polysaccharides and glycoproteins. The acid carboxypeptidase cathepsins ABCD acts on proteins. Then the beta galactosidase acts on these keratin sulfates, like or the galactosaminoglycans. Ceramidase acts on these sphingolipids like ceramide. Okay. Example, at least you should give 4. And if the question is specifically on the enzymes of the lysosome, then you should probably can give the maximum number of questions which you or the examples which you remember. Okay, so these are the questions which have already been asked on this. Okay, although there wasn't any question on the lysosomal diseases, still I will share 2 or 3 slides on these lysosomal diseases. Okay, so although you will find lysosomal disease in some of the notes of the classes, but I don't think that that is the question which is going to be asked anywhere soon, because it is not have been asked previously. Okay, because we have already almost 22 years of questions, and I don't see any questions apart from the structure, origin, function of the lysosome. Okay, so we will stick with these 2 points only, and I will share a separate 2 or 3 page one slider in my private Bhosle zoology group. So, and we will discuss all these questions in the live session, which will be going to be conducted tomorrow morning, 9 o'clock. Okay. So, have a great day. Let's meet in the next live session. Bye bye.